Okay, then welcome to this lunch seminar. My name is Gunnar Schulte. I'm professor at the Karolinska Institute, the Department of Physiology and Pharmacology. I'm a receptor pharmacologist, and that's the reason why we'll talk about ligands, receptors, pharmacology, the prospects of human therapy, but also windmills. And the last term obviously is supposed to symbolize the idea of Don Quixote fighting windmills, and the windmills in my case are biological obstacles, technical problems, editors, reviewers, the field of that puts some pressure on you for selection of getting the best results published and actually digging into the truth. This is the popular scientific title of the talk, but I also have a more scientific title of the talk is Understanding GPCRs and the GPCR nature of frizzles as a basis of drug discovery. And GPCRs are G-protein coupled receptors, a big target of pharmaceuticals. Adrenaline receptors, dopamine receptors belong to this group of receptors. And frizzles are the receptors that I'm focusing on, which I would like to talk about today and introduce you to my journey over the last 15 years to become a professor at this department. So first of all, who am I? Well, I'm a biochemist. I have my education from Germany, from the Free University in Berlin. And I came to Sweden in '96, starting small projects at the neuroscience department and then getting into GPCR pharmacology with my PhD project. And after some excursions to different departments at KI and a postdoctoral period at Monash University in Australia, I came back to Sweden and started to become independent. And one of the key experiences was a meeting with Martin Lohse, who, when I was looking into what I would like to research on, was telling me that you have to focus, you have to find a research niche, and if you really want to work on these weird receptors that are called frizzles, then you need to become Mr. Frizzled. And that was in 2006, and now we're here, 2019, and I think nowadays I would be able to call myself as an expert in frizzled pharmacology, and some people actually call me Mr. Frizzled. So over these years I had a long-standing interest in targeting these receptors pharmaceutically and therapeutically. The lack of knowledge prevented us from doing that, and only during the last 2 or 3 years we've gotten closer to actually being able to find drugs, to screen for drugs that target these receptors. And I would like to share parts of this journey with you today. So GPCRs, G-protein coupled receptors, are well-established targets for pharmacology. I guess in your education you encounter that you can use drugs to target adrenaline receptors, muscarinic receptors, or serotonin receptors. Maybe some of you take antihistamines during hay fever for allergy treatment. So GPCRs are validated targets. There are many GPCRs in the human genome, about 800. And here you see the class A of GPCRs, which includes adrenergic receptors, dopaminergic and serotonergic. And these red circles mean that they are generally targeted by validated drugs in the clinics. There are other groups of receptors that are not targeted, as you see here, and I would like to zoom into this part of the circle, which is the class frizzled. These are 10 receptors, frizzled 1 to 10, which generally bind big proteins that are ligands called Wnt, and a smoothened, which you might have heard of, which signals through hedgehog proteins and mediates hedgehog effects. So this is the family that I'm focusing on to my research. And the overhanging aim is to understand these receptors on a molecular level, to find drugs to target these, and then hopefully to promote that concept to clinics in order to be able to target these frizzles, these receptors, for example, for cancer therapy. I've taken along, even though smoothened is not really the focus of my research, I've taken along smoothened because that is a validated target used in the clinics for the treatment of basal cell carcinoma with small molecule drugs. But also other members of this family, like the frizzles, frizzle 1 to 6, I've listed some examples here, are involved in the development of breast cancer, of pancreas cancer, of gastrointestinal cancer, liver cancer, and many more. So the idea is to, again, understand these receptors molecularly and to target these by drugs. It is not only cancer that could be treated, there are many different indications that could benefit from my research. I have mentioned as well that one could, for example, treat lung or heart fibrosis using drugs targeting these receptors. So what are we doing? We try to, on a biochemical level, understand receptors and their pharmacology. So receptors are embedded in the plasma membrane of cells, and they are dynamic molecules that interpret a message coming from outside of the cell and that is transduced to intracellular communication. In the case of the frizzles, the ligand, the proteins that activate these receptors are called Wnt. They are big proteins, and they have a fatty acid modification that makes them lipophilic. And we try to understand the interaction between the receptor and the Wnt proteins. We're also trying to understand mechanisms of receptor activation, and it is basically how these receptors change their form during the activation process. Furthermore, we then, after this activation of the receptor, study how these receptors initiate communication in the cell and how they actually feed into different signaling pathways. And again, this molecular understanding is then important to target the receptors by drugs, for example, interfering with the binding of the ligand, with the activation process, or with the initiation of the signaling in order to prevent over-signaling, for example, in tumors. The over-signaling through these pathways, through Wnt frizzle signaling, leads to increased cell proliferation or metastasis and can then preferentially be inhibited by drugs on the drug in this pathway. That is the idea that which I come back to in the end of the lecture, that we try to build up screens that lead to understanding how we can drug these receptors with small molecule ligands. When I started, the overall overhanging question in the field was if these frizzles, even though they are classified as GPCRs and they look like GPCRs, if they actually are G protein coupled receptors. And this was a basic question in order to really understand the molecular mechanisms of this receptor activation, which then in turn is important to target them by drugs. So I will tell you now over the next minutes how I address this question and how we actually answer this question over several years of research. The timeline, my personal timeline, started somewhere, I let it start at the end of my PhD in 2002. I became a docent in pharmacology at the Department of Physiology and Pharmacology at Karolinska in 2008. And that's why I'm standing here today. I'm a professor in receptor pharmacology since some time ago. In the beginning, we were looking at Wnt signaling, Wnt-induced changes in brain macrophages, immune cells in the brain that are important during stroke or during brain tumors, and that are consistently surveilling the immune status of the central nervous system. Over the next years, on the coming years, we were publishing different papers on the central question, are frizzles indeed G protein coupled receptors? And that is a very abstract, very basic concept, but as I said, important for drug discovery ideas in the future. And we were systematically going through different frizzles indicating that they might actually be G protein coupled receptors. And last year, or this year actually, we published a paper where we discovered a molecular switch, which is an activation mechanism of these receptors on the movement of receptors. And I put this up also to refer back to the windmills that you face. You see the publication of that study was done in 2019, but the original discovery was done in 2013. So things take a long time, and many of the research progress that we do is actually dependent on technological development and concept development in the field. One of the concept developments is also a concept of efficacy, which is a general pharmacological term, which developed over many years, also in parallel to my studies, from a receptor being activated by a ligand, either not at all as antagonists do, or fully by agonists. But it has also been clear that there are ligands with a negative efficacy, so that an inverse agonist which actually reduce the activity of a receptor. And that will be important in the later part of the talk. And also, since I was talking about this drug discovery, we are now in the phase that we actually try to develop assays and run first simple screens to find molecules, and that has been firstly done in 2008 in my group. So the class Frizzled is composed of 10 receptors, Frizzled 1 to 10. They are all encoded by different genes. We don't understand how they are different really. They encounter 19 different Wnt proteins, which are ligands that are expressed in different tissues over development. And Wnt activation binding to the receptor and signaling downstream of the receptor is very important for defining top and toe of the body axis during development, for example. The head is defined, where the head is defined by these signaling pathways. Cellular proliferation and stem cell proliferation is defined by this signaling pathway. Intracellularly, we look mostly at these heterotrimeric G proteins that are activated by Frizzles, if they are G protein coupled receptors, but also to scaffold proteins that are called disheveled in this case. And these are two branches of signaling, G protein branches and disheveled dependent branches. And we are interested in how the receptors can actually differentiate between those two choices that they have. This is the whole family of receptors. You see that there are different colors that represent homology clusters. Some receptors are more similar to each other than others. So 5 and 8 are very similar to each other, 3 and 6, 1, 2 and 7, 4, 9 and 10, and smoothen is a bit separate. And I will come back to the individuals at some point. What I would like to do now is to ask the questions that I have asked myself over the last 10-15 years and provide you a bit of the idea how I went along to answer those questions. First of all, if these receptors are G protein coupled receptors, then they should, in normal cells, in cells that have endogenously Frizzles, mediate a physiologically relevant response through G proteins. And the first thing that I was doing in my young independent lab is to establish the isolation of primary mouse microglia cells, which are the brain macrophages, and we were stimulating them with Wnt proteins. And you see here that there's a signal induced from unstimulated to stimulated, and that is completely blocked by PTX, which is pertussis toxin. It's a bacterial toxin that blocks heterotrimeric GI signaling. So this is basically the proof that Wnt-induced signaling is blocked when you block heterotrimeric G proteins. This is just a quantification, but you see also here that this shift in band of disheveled, which is the other branch of signaling, is not affected by pertussis toxin. So some idea in the signal initiation is that disheveled signaling is not dependent through on G proteins, but G protein signaling to this MAP kinase pathway, which is sensitive to G proteins. Also, we could show that G proteins are not only required, but they're actually also activated. This is a biochemical radioactive activation assay of G proteins in these microglia cells, and we see that Wnt5a is actually inducing the activation of G proteins, and that this leads to a decrease in cyclic AMP, one of the basal G protein coupled receptor mediated signaling pathways. So the signaling part was clear here, Wnt-induced G protein dependent signaling, but is this actually physiologically relevant? We were looking then at pharmacologically or at immune factors on these macrophages, like inflammatory markers such as Wnt-induced protein expression of INOS, COX-2 and TNF-alpha, which are pro-inflammatory markers. But we were also looking at proliferation, and you see here that the proliferation was induced by Wnt, but it was reduced by the inhibition of pertussis toxin in this case. But it was also reduced by the inhibition, by pharmacological inhibition of the MAP kinase, which is this signaling pathway that we were looking at before. And the proliferation was reduced by this, but also an invasion. So when immune cells, macrophages are activated, they surveillantly invade into the tissue to check what is actually wrong in the proximity. And this is an invasion assay showing that cells that are on top of a collagen block will invade when they are stimulated with Wnt5a. And interesting enough, this was blocked by this inhibitor that is blocking the G protein dependent MAP kinase pathway. So the concept of this and the summary of this is, first of all, we can check the question. Yes, frizzles are signaling in a physiologically relevant manner dependent on heterotrimeric G proteins, because the Wnt stimulation of these primary mouse microglia would induce changes in the cell, molecular changes, but also physiologically relevant changes that are G protein dependent. So the pro-inflammatory profile of these receptors, of these cells, was changed by stimulation with the Wnt proteins. So that was one of the basic ideas. So if this is happening in real cells, then it's relevant, and then it could also be targetable in disease. So the next question I was asking myself is, do frizzles interact in a Wnt-dependent manner with these heterotrimeric G proteins? And this is just a physical interaction that we would like to measure. This is very tricky. We could not do that in microglia cells. We had to translate our ideas to more model cell systems which express fluorescently tagged receptors. This is because many cells express endogenous receptors where the tools were lacking to look at these individually. So the fluorescent tag on a receptor would enable us, here in this case the green fluorescent protein, would enable us to look at this frizzled 4 in this case individually, even though there's in the background expression of other frizzles present. And this assay is far too complicated to explain it to you now. I just want to make the point that we co-express two different proteins, a G protein that is red and a receptor that is green, and we're able to, by an assay that is called dual color fluorescent recovery after photobleaching in the microscope, to assess the interaction of these proteins. And we have done that systematically for different receptors. We have systematically done it for frizzled 4, 5, 6, 7 and 10. So not all of the whole profile, but we could see that there's a selectivity of the individual frizzles to different heterotrimeric G proteins. You might know that there are different ones. There are GS proteins, stimulatory proteins, GI, GO, GQ, 12 and 13. And for example, we will see clearly that frizzled 4 was signaling to G12 and 13, whereas frizzled 5 was only going to GQ and frizzled 6 to GI and GQ. So there was a selectivity profile of the frizzles towards heterotrimeric G proteins, but we were only looking at the basic interaction between these proteins. So again, we can check the question. Yes, these receptors indeed act through heterotrimeric G proteins by interaction, and that is symbolized by these blue blobs here that stand for the heterotrimeric G proteins interacting with the receptors. And we will also see indications that when we stimulate with the Wnt protein, that this complex of the G protein and the receptor was dissociate in order to initiate signaling intracellularly. And this fits very well into an old concept that was developed in the late 70s and the 80s, which is called the ternary complex, that has been developed for adrenergic receptors, where a ligand, adrenaline, binds the receptor, and that is promoting the association of the G protein in a high affinity complex. Again, very complex and very complicated, but the point that I want to make is that the classical G protein coupled receptor, the pharmacological principle, is that a ternary complex is formed with a ligand, a receptor and an intracellular partner. And that binding of the intracellular partner will affect the extracellular binding partner, and there's an allosteric interaction between these molecules through the receptor. And that is defined by a form of the receptor. because the conformation of a receptor can be inactive or it can be active. And in the active state, you can stabilize it with a ligand and with a G protein. And if we can show that, we have the ultimate proof of Frizzels being G protein coupled receptors. So then, to continue that, the next question that I asked myself was, do Frizzels react to Wnt binding with activation of the receptor itself, but also activation of heterotrimeric G proteins? We had shown in the microglia cells that Wnt stimulation would lead to activation, but we were there not really proving that Frizzels are involved. So that was the next step, and this feeds back into the idea that we can measure a conformational change of the receptor. So you have seen that these G protein coupled receptors, they have 7 helixes that span through the receptor, through the transmembrane domain. And it has been established for different receptors. That's why, for example, Brian Kobilka and Robert Lefkowitz received the Nobel Prize in 2012, because they could show these activation mechanisms for example, adrenergic receptors. And if the same concept would be true for Frizzels, that this helix 6, the TM6, would actually swing out and open and give place for the G protein, then we should be able to measure it, again in a very complicated method that is called FRET, where we measure a change in the localization of a fluorescent protein here and a fluorescent moiety here in the receptor. So if these two parts move apart upon stimulation of the ligand, then we should be able to see that. And in fact, that's true when we build such a probe for receptor activity. We see when we give Wnt ligands that there is a decrease in FRET, which means an opening of the receptor, arguing that stimulation of the ligand will activate the receptor. This can be done on the microscope, this can be done on a plate reader, and the idea is that activation of the receptor would then lead to an activation of the G protein that is just downstream. And there we can use similar methodology, just that the fluorescent proteins are not on the receptor themselves, but on the G protein. Here we have used a methodology, it's called BRET, which is based on a luciferase and a GFP moiety on the G protein. So if the activation of the G protein is associated with a dissociation of the G protein, then we should see a decrease in the signal. And that is the case when we have this G protein activity probe expressed in the cells. With Wnt stimulation, we see a decrease of the signal only when we have Frizzled-5 present in these cells. So again, a proof that Frizzled-5 presence would mediate G protein activation. Again, we can do this on the microscope, and we can do this in a plate reader, and we can actually do a concentration response curves with Wnt, which is some of the first really pharmacological data that we could be able to obtain. Again, we were not completely satisfied by that and would like to have a measure for this activation of the receptor, also digging deeper molecularly on the mechanism of activation of these receptors. For this, we would use something that is an engineered G protein, again with a yellow fluorescent tag, and a receptor that is luciferase tagged and engaging into these BRET assays. And the idea here is that the G protein is a measure of the conformation, active conformation of the receptor. And when we do those concentration response curves, we see that the wild type receptor is responding quite modestly, but then when we mutate the receptor in a residue that is in this helix that is swinging out, then we get a huge response and an increase in the response. And this residue was at the same spot where mutations are present in cancer that lead to, when they are mutated in smoothen, that lead to basal cell carcinoma, an oncogenic mutation. We were able to define that for the whole class of Frizzels, finding a general activation mechanism by identifying this, what we were calling the molecular switch that is responsible for activation of the receptor. So there again, we can check the question, Frizzels indeed react to Wnt binding with activation of heterotrimeric G proteins, but also with, as I said, conformational changes of the G protein, the receptor activation. and the dissociation of the G protein. I've introduced the idea that I'm a receptor pharmacologist to understand receptors, but that is not the sole aim. The aim is, of course, to use that understanding to find new drugs. And that's then, of course, the next question which we honestly can address with the success of the last few years. And I would like to start arguing for that with one of the figures of our last paper, where we actually identified by genomic screening or genomic analysis of data of cancer variations in patients and comparing them with the natural variations in the normal population, showing that the highest mutated residue in all frizzles in tumors is this little residue on helix 6, the arginine that I was introducing before, that is leading to an opening, that is promoting an opening of the receptor, an activation of the receptor. This residue is not mutated in normal population, in people, in the normal general population. And if you make a ratio of these two data, you see that this residue in the helix 6, highlighted here in red, is really the only one, the highest mutational hotspot in this family of receptors. This argues that it's important for pathology. We have not proven that it is causative for the pathology, but it tells me that if this residue is involved in receptor activation, then targeting receptor activation pharmacologically would be a good idea for the treatment of cancer. So, and I think we can exploit our findings and the knowledge that we gained by for finding new drugs. We would be able to find drugs that interfere with Wnt binding to the receptor. That is partially ongoing, that people have designed antibodies that compete with the extracellular Wnt binding to the receptor. But I think it's more promising to actually design and find small molecule ligands by drug screening to affect the conformational changes that we were showing in these receptors. To indeed act on the core of the receptor, preferentially as inverse agonists, which would reduce the activity of the receptor, or as antagonists, to treat tumors that present with high levels of Wnt or mutated receptors or high levels of frizzles, which would be proliferative or tumorigenic. The drug screens that we proposed and we try to invent are monitoring conformational changes, G protein recruitment and G protein activation. And we try to use the methodology that I was describing to you, which has been made possible by recent technological events in the field. So with this, I would like to finish to say that gaining knowledge in receptor pharmacology, ranging from physiological cells to molecular details on receptors, will not only satisfy curiosity, but will also lead to a progress in pharmacology and potentially in therapy. The way is long to a therapy for cancer, obviously, but I think we are on a good way. So I've answered my favorite questions in the field over my career, and I would like to then finish with that I've told you that there's a ternary complex, there's a ligand, there's a receptor, and there are G proteins. And if we understand that molecularly, then we will also be able to exploit that knowledge for drug discovery and assay development. Class F receptors, frizzled and also smoothened, are classified as G protein coupled receptors. I think over the years we have shown that they are G protein coupled receptors. They behave especially like this is the GLP-1 receptor, like a normal GPCR. But they're also different, and we have not really grasped the differences and particularities, how they act special. For example, through this disheveled protein that I've mentioned initially. I think there's a potential of targeting the receptors in a biased way, that maybe the G protein over the disheveled pathway could be modulated, but that is still music and ideas for the future. But I would like to finish with the idea that Frizzels, yes, are GPCRs, and that is a finding itself, and that we can engage in drug discovery. Now with the knowledge that we have in a structural, guided manner, but also in a mechanism-based manner. And with this, it only remains me to thank you, of course, for your attention, but also to thank the people that have been involved. As I told you in the beginning, I came from Berlin as a young biochemistry student, and I was mentored by Professor Gunnar Grant at the neuroscience department, which was a very exciting time scientifically and privately for me to somehow put the grounds for me to stay in Sweden. And I did a PhD thesis with Professor Bertil Fredholm at the Department of Pharmacology, entering GPCR pharmacology with work on adenosine receptors. And Professor Ernest Arenas at the MBB was my first contact with the Wnt field and Wnt signaling, looking into the development of dopaminergic neurons. But also the people who I've been working with over the last years in my lab, and these are it's the chronological order of the PhD students I've supervised to the thesis defense. Number 10 is in work, and I really like to thank those people who are essential for getting the concept done and hopefully engaging into future development of future therapies. Thanks, and I hope you have questions.